Okay, good morning, everyone, and welcome back to our third and final day of the 2023 Invasive Species Forum. Before we begin, our session is going to start at 9 a.m., but we have a special guest this morning and a couple of announcements to get through. So before we begin, again, we greatly appreciate the significance of the lands and waters and all living things and offer our gratitude to the Indigenous people for their care and teachings about our earth. Our relationships with Indigenous communities are important, and we will continue to listen and learn how we can be in a good relationship with Indigenous peoples, the lands and waters, and all living things, and act accordingly. The Invasive Species Center acknowledges that our head office is located on the traditional lands of the Anishinaabeg, the Batchewana and Garden River First Nations, as well as the longtime settlement of the Métis people in the Robertson Huron Treaty area. We encourage everyone to take the time today and every day to consider the traditional lands on which you reside. It's my pleasure to welcome our special guest of today, welcoming remarks from Parliamentary Assistant of the Ontario Minister of Agriculture, Food and Rural Affairs, PA Trevor Jones. Thanks for being with us this morning, PA Jones. You can take it away.
Hi, good morning, everyone. Thanks for having me this morning on this cold, wet morning. I'm Trevor Jones, MPP for Chatham-Kent-Leamington and Parliamentary Assistant to Lisa Thompson, our Minister of Agriculture, Food and Rural Affairs. And I'm so pleased to join this meeting today. I know how important this event is. It enables us to share best practices, gather information, and bring needed attention to invasive species, issues, research, prevention, and management. Over the course of this meeting, we're fortunate to have a number of experienced speakers presenting, including Omafra's own Wendy McFadden-Smith, Horticulture Integrated Pest Management Specialist, who will share key insights. The Invasive Species Center's commitment to preventing the introduction and spread of high-risk invasive species in Canada is now important as ever. Invasive species threaten not only Ontario's natural environment, but also our agricultural sector and our food supply. In our province, our agri-food sector is essential to a strong and prosperous economy. It also supplies stable, close-to-home jobs for 1 in 10 Ontarians. To protect our environment, our farms, and our food, we must collaborate, stay proactive on invasive species research, policy, and program development. Our government is happy to deliver on this. For example, we work continually and closely with our Ministry of Natural Resources and Forestry on early detection and prevention of wild pigs throughout Ontario. We're also staying on top of invasive plant species. For instance, I know that Phragmites can be of particular concern to farmers and rural municipalities, like my own communities of Chatham-Kent and Leamington. These Phragmites outbreaks can sometimes invade entire agricultural plots and affect municipal drains. Phragmites are an interesting grass species that have a unique ability to drain water effectively, but their thirst also changes the entire wetland habitats where they're sometimes found. And so this is something that's very common in Leamington and throughout Chatham. And it's something that my wife, who's a high school science teacher, teaches her high school science students, where those beautiful cattails are no longer around filtering our clean water, but the Phragmites actually drain it because of their thirst. So that's very relevant to me. Our ministry is also working with other levels of government, the ag sector and the Invasive Species Center, to identify strategies and implement actual response plans for invasive species, such as the spotted lanternfly. So if they do make their way to Ontario, we're prepared. It's clear that this important work is integral to our vitality and prosperity in our ag sector and our environment. Please keep up the outstanding work you're doing. Work together and work for the environment and our farming sector throughout Ontario. Thank you again for inviting me to share a few words today and some of my experience. I wish you the very best in this forum. Thank you for the work you do.
Thank you so much, PA Smith, for your wonderful welcoming remarks. A great start to the day, for taking some time with us this morning. We actually have some great talks set up for today on Phragmites, so you've really set us up well for the day.
I've had to actually I've had to jump in a ditch and harvest it for my wife's science class back when she taught high school science in Windsor. And
that's so great.
she started my learning journey. And then her students, I was invited to kind of share into some of their forums in class. Their students taught back. And it's amazing their aptitude for learning and their ability to kind of, understand and take a link to that and see it in their communities and really be interested in what the impacts are. So thank you. It's
Yeah.
very relevant and we appreciate it.
Absolutely. Thank you so much, PA Jones. Again, thanks for being here with us this morning. I know you've got another meeting to get to for a busy day, so we'll let you get going.
Thank you, Mackenzie, and thank you, everyone. Have a great day.
Bye. Okay, everyone, so we're a little bit early for our 9 a.m. So I'll give you, just as we have the last couple days, a 10-minute break or so to get your coffee and get yourself ready for a good morning of invasive. So looking forward to seeing you. We'll meet back in about 9 minutes at 9 a.m. Okay, welcome back, everyone. It's about 8:58, so we can get ready to start our day here as people kind of trickle back in. I just have a couple of housekeeping items to go through this morning before we get started, especially if there's anybody new joining us for the first time this morning. My name's Mackenzie, and I'm going to be your MC through the rest of the day today. I'm going to introduce our wonderful moderators that are helping out to go through the sessions with each of the speakers. Please use the Q&A feature for any questions, and your session moderator will take you through those questions at the end of each talk. There will be some ISC staff monitoring the chat for any technical issues. So if you're having any audio issues or any other, please just put that in the chat and we will get to you as soon as we can and help you out. You can also refer to the attendee guide that should have been distributed through email prior to the event. We have frequently asked questions in there that will hopefully address any of your issues. We have a keynote plenary again today at 12:30 Eastern Time. Join us this afternoon with Bethany Bradley as she speaks on integrating climate change into invasive species policy and management, invasive plant policy and management, sorry. And one more item, we'll be distributing a brief survey at the end of the forum, and your feedback's really important to us. We have some new events that we've added to the forum this year, so we'd really like your feedback on them and how things went this year. We do use your feedback every year to hopefully improve our event, so definitely take a couple minutes to fill that out for us. So without further ado, it's 9 a.m. right on schedule. Let's get started. I'd like to introduce our moderator for the morning, Sherise Vincentini. But before I pass it over to you, I do have just a couple of announcements for things going on today. So we do have trivia at 11:50 a.m. So come showcase your invasive species knowledge for a chance to win a prize. All you need to do to join the trivia session in Zoom event is just join the session in Zoom events and have your mobile device handy. So looking forward to that. And just a congratulations to all of our annual award winners that were presented at last night's virtual reception. All the winners and nominees demonstrated excellence, innovation, and leadership in invasive species prevention and management. So you can check out the ISC's social media for a recap of the evening. With that, I will pass it over to Sherise. Sherise is the ISC's community action leader. He puts together some really fun and practical projects that bring community scientists into the world of invasive species, like our community science tree check forum and her work connecting youth through a local insectarium. Also enjoys giving presentations about invasive plants to community members, so she's going to be great at taking you through this morning's session on developments in invasive plant research.
Thanks, Mackenzie, and good morning, everyone. Thank you all for joining us this morning. I'm very excited to introduce our speakers for this session on invasive plants and development and research. As Mackenzie mentioned, just a reminder, if you do have any questions throughout the presentations, just post them into that Q&A function, and I'll be reading those out at the end of each presenter's talk. So you can hold on to those until the end. Okay, and to start us off this morning, I'd like to welcome Sherise Vialva from Queen's University to talk about the effects of climate warming on competitive interactions between Alliaria, I'm not very good with Latin names, Alliaria petiolata and Vincetoxicum rossicum. So I'll welcome Sherise to the stage, and I'm sure she'll do a much better job at pronouncing the Latin names.
Can everyone see my screen?
Yeah, right now we can see it in PowerPoint. You just need to start the presentation.
Okay, and I can't promise I'll be able to pronounce the Latin names any better.
Excellent, it looks perfect. Take it away.
Okay, so hello everyone. My name is Sherise Vialva and I'm a second-year master's student from Queen's University. And I'll be presenting my research today, exploring the impact of climate change on the competitive dynamics between Alliaria pallidea and Vincetoxicum toxicum. So biological invasions in a warming climate. Mean temperatures have a 90% probability of increasing in Ontario by 2 to 4 degrees Celsius between 2020 and 2049. This is a huge increase in temperature for such a short amount of time, and it's currently happening right now. Given this, I want to know the answers to these key questions. How will future climate warming affect invasive species spread, abundance, and fitness? And could interactions between invasive plants alter invasion trajectories in response to future climate warming? So my research focused on these two study species, Vincetoxicum rossicum, also known as dog strangling vine, which is a perennial vine, and Alliaria pallidea, which is garlic mustard, and it's a biennial plant. And to note from here on, no more Latin names. I'm going to be using the common names. So they're both originally from Eurasia, but have been in North America since the late 1800s. This may seem like a long time for people, but on an evolutionary scale, this invasion is quite recent. And as you can see, even though they haven't been in North America for that long, they have had a huge impact on the local ecosystems. Both species have a tendency of creating dense populations of mostly themselves and exclude many native plant species from the areas that they invade. This is seen even regionally. As you can see in Southern Ontario especially, they are very common, and there's lots of population overlap, and therefore lots of potential competition between the two. So to answer my questions, I conducted an experiment at Queen's University Biological Station, also known as CUBES, during the spring and summer of 2022. My experiment had a setup of 20 1 meter by 1 meter plots. And as you can see, half of the plots here had open top chambers built on top of them to passively increase the temperature by 1.8 degrees Celsius to simulate climate change. We use soil probes to measure the differences in temperature in the soil in all the plots. And we measured plant height, number of leaves, and plant area as a measurement of plant fitness. In addition, we also had a competition treatment. Each plot was subdivided into 3 subplots that had either garlic mustard alone, dog strangling vine alone, or both grown together. So going into some of the results, the probes that were measuring the temperature showed that the chambers did work. There was clear differences between temperatures between chambered and non-chambered plots. On average, the chambered plots were about 1 degree Celsius warmer on average. So going into how the plants reacted to this warming treatment and the competition, who benefits from climate warming in terms of height? First off, when grown with dog strangling vine, the warming treatment had no significant effect on garlic mustard height. However, when grown in a monoculture with just themselves, garlic mustard the warming treatment did have a really significant impact on their height. And for dog strangling vine, there was clear differences between the dog strangling vine that was grown with the warming treatment but how they reacted differed depending on if they were grown just with themselves or with garlic mustard. When they were grown by themselves, they were much taller, but when they were grown with garlic mustard, they were much smaller. In terms of number of leaves, once again, when garlic mustard was grown with dog strangling vine, the warming treatment had no significant effect on the number of leaves. But of course, when grown only by itself, the warming treatment had a significant effect on the number of leaves garlic mustard had. And it was much more than when it was grown without the warming treatment. And for dog strangling vine, there wasn't really much of a difference between individuals that were grown with or without the warming treatment, or with or without garlic mustard. And finally, leaf area. There was really no significant effect on leaf area for both garlic mustard and dog strangling vine. And this mostly held true regardless if they were grown separately or together. So in conclusion, climate warming may increase fitness for both species, which could lead to greater spread and abundance. Competition limits. We found that competition limits growth, especially for dog strangling vine. And the benefit of warming appears to be enhanced in monocultures. Just to note, there are some key limitations to this study that I conducted. First off, it was only focusing on first year of growth. We are planning on measuring the long-term survival and reproduction in the future. And another thing is that the chambers could only partially simulate climate warming. Climate change is very complex, so we could only really modify the temperature. So going into this, what are the impacts for future population control? It's likely that monocultures of garlic mustard and dog strangling vine may increase in abundance under climate warming in the future. As a result, areas with monoculture should be top priority for removal right now to hopefully prevent it from being a bigger headache in the future. And I'd like to thank my supervisors, Dr. Ecology and Dr. Andreas, my committee members, Dr. Yamanowski and Dr. Friedman, lab partner Andrew Lee, who helped a lot in preparing their experimental site, and undergraduate students Logan and Abby for also helping with the site preparation and data collection as well. And that's it.
Thank you so much, Sherise. That's a very interesting topic. We do have a couple questions already in the Q&A, so I'll go ahead with those. Firstly, Lauren asks, does garlic mustard and dog strangling vine overlap in their native ranges?
So yes, I don't know if I should share my screen again, but
Sure, you can go ahead.
So this map here shows it has data from GBIF and EDD maps, and it shows like a portion of Southern Ontario, and each of these dots is a reported instance of either dog strangling vine or garlic mustard. So they are very widespread in Southern Ontario, and it's very likely that there is overlap. not included in this presentation was that I also did a field survey during the spring and summer of 2021. It just couldn't fit in this presentation because it's only 15 minutes long. And I did find places where both species were growing together. So there is overlap between the two.
And what about in their native ranges?
In their native ranges, I'm not sure, but I do know they're both from similar parts in Europe, so it's very likely there is some overlap.
Okay, excellent. Thank you. Rob shares interesting results. Sorry, I might have missed this. How long were the measurements taken after setting up the plots?
So we started, after we set up the plots, we started taking measurements, I think like a week and a half to 2 weeks after the plants were established. We transplanted the seedlings for both species in the plots to have them both there. So we let them get established in the plots, and then we start taking the measurements over the spring and summer.
Perfect. Thank you. Sharon asks, are you considering researching which native species compete against these two?
Not really. Yeah, this photo here kind of shows that native herbaceous plants don't really compete very well with either species. But this is, of course, the most extreme examples I found during 2021.
Right.
There are other places where there were other species that were growing with them, but it wasn't as dense as this.
Okay, thank you. Lauren asks, could the plants be grown in a greenhouse setting to simulate higher temperatures?
Yeah, possibly. I know the average temperature was a bit lower than 1.8 that it was supposed to be, but that was averaged. The probes, they take 3 different measurements, one above the surface, one at the surface, and one below the surface of the soil. So those were averaged. I'm sure if we had separated them, we would have seen widely different temperature ranges between the chambered and non-chambered plots, probably especially for the one above the surface, but these were all average, so that could have made this average look a lot smaller than it should have been.
Yeah, absolutely. Thank you. Nadia asks, how deep into the ground were the dividers that separated garlic mustard DSV and then the garlic mustard plus DSV?
I would say maybe about 10 to 15 centimeters. So it was kind of difficult to put the dividers in. They were like lawn edging, like rubber plastic lawn edging that we cut up and put into the plots. So and then buried them. So it was kind of difficult to work with them. But yeah, I say at least 15 centimeters deep is how deep the dividers were.
Excellent, thank you. And then we have another question. David asks, any plans for how European buckthorn might influence growth and how soil nutrient change in relation to buckthorn presence?
So my focus was more on the above ground effects. So how the plants impacted each other's growth. And there is someone in our lab that's looking at how how dog strangling vine impacts the soil as well as garlic mustard, and what would happen when the two are grown together. So that's more of a question for them, but they're not here right now. So yeah, there are people in our lab that are looking into that.
Okay, is anyone looking into European buckthorn? Like along the same lines?
Oh, I thought that that was a different name for dog strangling vine. No, no one in our lab is doing anything on European buckthorn.
Okay, good. Thank you. We have a question from another David. Did you have any success killing either of them?
Well, we didn't really want to kill either of them because then we wouldn't have any data for this experiment. Personally, I have done polls of dog strangling vine. It's really tedious, it's really difficult, and it takes a long time, and you have to be very vigilant on it. So if you have like infestation of either species on your property, my best advice is to always pull every year and to keep an eye on it, because there would be seeds in the seed bank in the soil, and to plant native plants after you pull them to help prevent any seeds that are seeds of either species from getting established again.
That's excellent. Okay, thank you so much, Sherise. There is still a couple more questions coming in. There's one in the chat and a couple in the Q&A. If you wouldn't mind typing out your answers on the Q&A function. But in the interest of time, we're going to have to move on to the next presenter. Thank you so much, Sherise. Okay, next I'd like to introduce Audrey Plotkin from the University of Massachusetts, Amherst, to talk about the Northeast Risk Management Network, a replicable model for translational invasion ecology. And you can take it away, Audrey.
Okay, great. Do you see the full screen?
I do.
Excellent. Perfect. That's what I would like. All right. Well, good morning, everybody. It's great to be here. And I'm coming to you from Massachusetts, so south of the border, to talk about a really great organization that I'm part of, the Northeast Regional Invasive Species and Climate Change Management Network. Before I get into this, I'd like to acknowledge my co-authors, Jenica Allen, Carrie Brown Lima, Bethany Bradley, and Tony Lynn Morelli. And that this consortium is, a group of different organizations, including University of Massachusetts, the U.S. Geological Survey, the New York Invasive Species Research Institute, which is at Cornell University, and the Northeast Climate Adaptation Science Center. And I'm going to be talking to you, give you a little overview of the organization, and then really delve into some recent work that we've been doing on prioritizing range shifting species, and I'll focus on plants, and then a variation on that of sleeper species, prioritizing those. And then finally, make a pitch that we would love to have you join our work. Okay, so the Northeast Regional Invasive Species and Climate Change Management Network, were founded in 2016, and really to tackle the problem of the compounding effects of invasive species and climate change. And we do that by synthesizing science, sharing manager needs and knowledge, building stronger scientist-manager communities, and conducting some original priority research. And the model that we use to do this is what we like to call translational invasion ecology. So translational ecology is something that's gotten some traction in recent years. And so a couple of years ago, Tony Lynn Morelli led our team in writing a paper about what does that mean for invasion ecology. So it's really putting together researchers and managers and policymakers at every step of the way to identify problems, figure out solutions, come out with best management practices as well as peer-reviewed research, and then always looping back to figure out how can we do better the next time? What are the new questions that emerge as we work on this problem? And so I'll give a couple of examples of that in the focal parts. And that we're really excited that this seems to be a replicable model. So we started in 2016, and in the last few years, we've been seeing new regional invasive species and climate change management networks popping up. This map shows the different regions of the US climate adaptation science community, and there are emerging networks in the Pacific Northwest. The North Central Pacific Islands and the Northwest and Southeastern US. And there are new regional groups under consideration in the US Midwest and Southwest, as well as in Canada. So that's super exciting that, how we're organizing our work seems to be something where, although we have common concerns and information needs across these regions, having this at the regional level is really appropriate because there's variations in what the most pressing needs are in different regions and who the stakeholders are. Okay, so I'm going to delve into some recent work that we've been doing as a network. And the first topic is on prioritizing range shifting species. And this work started with Jenica Allen doing a really great study looking at what invasive plant species have the potential with climate change to expand their range, especially into the northeastern US, since that's our focal region. And as a result of that work, there's those results are shared on this excellent tool called EdMaps. The one problem with it is that it covers only the United States. It doesn't go into Canada, at least not yet. And the other challenge is that, with climate modeling and looking at the climate suitability for a variety of invasive plants, Jenica identified 100 potential invasive plants that could be moving into our area. And that's really overwhelming. So we took the next step by prioritizing that list of 100. And this is how we did it. We worked with some excellent undergraduates at the University of Massachusetts and developed a protocol using the IUCN's Environmental Impact Classification for Alien Taxa, or ICAT, assessment protocol. And what that does is you use existing scientific literature to hone in on a particular species, screen all the articles that are about them to pull out papers that measure their potential impacts on ecosystems, and then set up a way of giving those impacts a score from minimal concern to major concern. So May Rockwell Postel and Bridget Griffin started this with Southern New England and New York, and then a couple years later, Will Colville expanded that to Northern New England. And as undergraduates, they each got first authored journal publications out of that, which is awesome. And also we take a lot of the original research that we do and synthesize it into 2 to 4 page summaries that we call management challenges. And so this is showing a part of the management challenge that came out about these what these priority range shifting species for Southern New England and New York are. And so the nice thing about it is that we, kind of really summarize what the 5 out of that 100, what the 5 major impact species that we think are most likely to affect northeastern ecosystems are, so that we can suggest, hey, everyone, these are ones that you really should be keeping an eye out for and trying to do some rapid response and early detection work to, make sure that you nip anything in the bud with those. But we also expanded that to a larger list of additional species that managers, depending on what ecosystem they're in, or what their risk tolerance is, or what their capacity is to monitor for other species, ones that could have an impact, but we weren't as certain, or they didn't seem like they would be affecting, the most prevalent ecosystems in our region. So we included all of those as well in that management challenge. And we also made sure that this is a replicable research and management model by one of the products from this was a data template, so that if you're interested in trying your own ICAT assessment for different species to assess, how big of a concern are they? How big of an impact might they have? That's something where we put together a data template that you can download and use yourself. And I'll just mention that, I'm focusing here, especially because we're in a plant session, on the work that we've done with plants that has been the kind of core strength of our team as we've gotten started. But we really are also expanding from plants to invasive forest insects. That's something that's my research focus, and marine ecosystems. And I also wanted to take this opportunity to highlight that one of the other regular research synthesis works that we do is, in addition to putting together, maybe 2 big synthesis management challenges a year, we have every other week we send out a research summary of a relevant primary literature article that looks at some aspect of the interactions of climate change and invasive species, give a little summary, and then just like some really simple bullet points of take-home points and management implications. So that's something that at the end, I'll invite you to join our listserv, and that's the kind of core thing that you would get from that, is this every other week kind of research digest. Okay, so I've been talking about range shifting species. Kind of related to that, or kind of a cousin to that topic, are what we call sleeper species. So these are potentially invasive species that are established in an area but are not spreading because there's some sort of biotic or abiotic limit to them. And for example, the climate is too cold for their populations to really get going and expand. This is something that we introduced in a management challenge in 2018, where we introduced the concept. And this also just, this is just a little screenshot of the 2-pager to give you a sense of what that product looks like. So here we really, got into the idea of sleeper species and why they're important. There's something that's, that's there, but not maybe on your management radar because they aren't invasive yet, but gosh, that's the right time to try to control them because there aren't that many, right? And at the end of that management challenge in 2018, we, also came up with the idea of, okay, the next step is that we really need to prioritize sleeper species because, gosh, there are a lot of non-native plants that are established, but not yet invasive. How do you decide which ones to worry about? So we suggested that you identify the pool of naturalized sleeper species, assess their potential invasion and impact, and then, of those, really figure out, well, which ones can we, make a difference? Which ones are possible to eradicate, and focus on those. So that was our suggestion, and that's exactly what we did over the last couple of years. And that this work was led by a master's student, Ayo Uhuru. who put together her master's thesis as well as a management challenge on prioritizing sleeper plant species, again, for the northeastern US. And so in addition to putting together some examples, for example, everyone's favorite kudzu, which does seem like it is, it is present, but not invasive in some parts of the Northeast. It has the potential by mid-century, those orange areas are places that the climate would be suitable for it to expand its range by mid-century. And so really, with climate change, we expect this, that kudzu is very likely to start spreading, wake up from being asleep, and really have a big impact. There's also, you're welcome to, one of the other things that IO put together was a full database that summarized the ICAT assessment, just like for the range shifting species, assessing what sorts of impacts these different plant species might have and how major they can be and what counties they are currently found in. And so you're welcome to download and explore this whole database of 179 species.
You have 2 minutes, Audrey.
Thank you. Okay, so gosh, a lot of, thinking about invasive species and then throwing in climate change, that's really daunting, but there is a silver lining and let's use it. Climate change offers us great opportunity to be proactive about invasive species management. And I think this is something that Bethany Bradley is going to be talking about in the keynote more later this midday. So some of the things that you can do is, learn from your neighbors to the south of what eradication and management techniques have worked and be ready to implement them. We've learned so much from our neighbors to the south, and we're really excited to pay this forward to our neighbors to the north. And finally, there's lots of things that you can do as invasive species researchers and managers. But I think what's really special about our network is that it really brings in that climate change interaction. So we invite you to check out our website, which this will take you to kind of a homepage where you can look at all the different regional risks. And you can sign up for bi-weekly research summaries. And finally, just next week, we are having our annual symposium next Tuesday and Wednesday. It's all virtual on Zoom, and we invite you to register and join us. So thank you so much.
Thanks, Audrey. That was very interesting. I loved learning about the whole network itself. I mean, I personally see a lot of opportunity to engage the public on this too, especially, once species might be identified in the sleeper species, under sleeper species or potential sleeper species, and, get the public to start at least reporting those, whether it's through iNaturalist or if they're not on EdMaps because they're not identified as invasives yet. Or even to help identify potential species that they might be worried about in their communities. I see a lot of potential there. Are you looking into, or is this kind of open to the public to join the network, or is it more for professionals? Do you have that community science component?
Yeah, so we certainly do. It's not been a super strong emphasis of what we've done so far, but we certainly have, interested members of the public who, come to our webinars and symposia, and certainly that's something that we could, probably through some of our manager partners, implement more. Yeah.
Oh, that's excellent. Very exciting. Okay, I do have a question. Oh, no, a bunch of thank yous. As I read them.
Okay.
If anyone has any questions, we do have a couple more minutes. And one thing I would like to note is that EdMaps is actually in Canada now.
Oh, awesome.
It's been in Ontario and Alberta for a couple years. But more recently, we've been trying to expand that, and it's now a national platform, so anyone can report to it from anywhere in Canada. So you should be able to have some more data.
Well, that's great. Yeah.
I do have a question from Sharon. What advice do you have to try to convince municipal governments or others of the need to put some resources to this? We're a conservation group and trying to get them to understand that it will be cheaper in the long run to deal with it now. And it's like pulling teeth.
Yeah, that concern is what we hear all the time is, it's just so, when you see what's on fire right now, that's where the resources go. And so I think it's invite those municipal governments to learn a bit more. One of the things that we're trying to do is do some more regional coordination, for example, of invasive pest councils at the provincial and state level to really advocate proactively listing species and trying to coordinate that. So I think hopefully that sort of momentum will start to convince, but I think that being reactive rather than proactive is just kind of human nature. So yeah.
No, that's very true. There are a couple other questions and a couple people asking for links both to the database that you shared and to newsletter and the conference. If you wouldn't mind going to put those into the chat and answering the question that's in the Q&A box. But in the interest of time, we're going to move along. Thank you so much, Audrey. Very interesting.
You bet.
Okay. So next, I'd like to welcome Dan Buonaluto from University of Massachusetts Amherst to talk about the phenological response to climate mediate seedling competition with an invasive woodland herb. And with that, I'll let you take it away, Dan.
Thanks so much. And thanks to everyone for tuning in. I'm excited to talk a little about this work on phenological responses to climate and how that affects competition between invasive and native species. And this is work that I did as part of my PhD in collaboration with Dr. Lizzie Walkovich at the University of British Columbia. And I don't know if she's here right now, but probably she would point out that I just committed a sort of talk no-no in that I introduced a potentially term of scientific jargon right in the title slide, phenological response. So just to get started, I want to give a kind of working definition of phenology for us, which is the timing of seasonal life cycle events. So that can be things like migration time or reproduction time, but in plants, we really think about it as the time that leaves or buds or flowers emerge in the spring, or fruits disperse in the fall, or when seeds germinate. One of the strong associations between invasive plants and phenology is that invasive plants are characterized by early and rapid phenological growth. So they're the first species to green up in the spring. Their germination is often very rapid, and not so relevant here necessarily, but also they're often some of the latest species to go dormant in the fall. So there's a lot of observational work and also theory to explain why perhaps this is actually not just chance, that there is a sort of mechanistic relationship to their invasion success. So I'm trying to kind of scheme this out a little bit, but if you imagine, right, that competition is a race for, to acquire seasonal resources before your competitors, early phenology or rapid germination allow you to get a head start in that race and begin to draw down resources before your competitors. That's depicted here by the kind of driving tree. And also potentially modify the environment, create more shade or change soil characteristics that make it harder for your competitors to emerge. So this makes a lot of sense, but I think one of the challenges of really defining this as a mechanism, not to mention quantifying it, is that often these phenological traits, so these rapid germination and early phenology, are associated with lots of other traits that would make a species a good competitor. For example, rapid growth rate or high fecundity, right? So it's hard to really just from the observational studies isolate the mechanism of these timing differences. So experiments have come to bear to help us with this a little bit. And so there's a a common experimental setup that has been implemented mostly in California grassland ecosystems, as well as some desert ecosystems in the Southwest, where it's a fairly simple concept. You take two competing species, and as you see up here in the top panel, you plant them at the same time, and then you track their growth to the end of the season, and you kind of look at the difference in their growth rate. Then you take those exact same species under the exact same conditions, and the only difference you make in your second treatment is you plant one some period of time before the other. So imagine this is 2 weeks before, and then again, you look at the relative difference in their growth at the end of the season. When you compare the relative differences in these two treatments, you can identify how much that phenological advantage, as we call it, so that head start, mattered to the competitive outcome, right? And so this has been a sort of tried and true method that has really given us an understanding that there is this mechanistic relationship. But there are a few problems with thinking how to, the next step, which is to translate this experimental approach into like understanding real biological systems. So the first, right, is this amount of phenological advantage is a decision that the researchers are making, right? So it's unclear if this species here depicted in black ever actually germinates 2 weeks before the species in white in real systems. So and you could obviously change this effect size considerably, whether you decide to plant the species 2 weeks before or 4 weeks before or 6 months before, and those would really change the outcome of the experiment. So there's some kind of experimental bias introduced there. And the second piece is, while this may be a really appropriate idea in annual grasslands, in many of the temperate ecosystems that I'm going to focus on today, seed dispersal happens at the end of the growing season, so in the fall usually, and then there's a period of dormancy, and the resumption of growth in the spring, so the entrance to competition, happens less about when the species are planted and more how they respond to the spring warming and how they can emerge from dormancy. So that is a piece that I want to talk a little bit about today, thinking about how climate is going to be affecting these dynamics. So just as a kind of brief introduction, when we think about germination phenology and temperature, there's two main temperature cues that affect in many temperate species that affect their germination. So the first is an exposure to a period of cold stratification. So this is exposure to cool temperatures that usually occur in the winter or spring. And those temperatures are somewhere usually between 0 and 6 degrees, but that can vary by species. And that is required for many species to release them from primary dormancy. And then once that happens, the incubation temperatures, or the warm temperatures that they're exposed to in the spring, that will also affect the speed and completeness of their germination. So I wanted to potentially try to use these, the fact that species may have different responses to these two variables to see if we can create an experiment to test the effect of phenological advantage on competition. So kind of this is an experiment that has really two questions. One is that, do the differences in species responses to temperature variation, does it drive sufficient variability in relative germination timing to potentially matter, right? Do we see this kind of variation on the order of weeks in phenological advantage just based on temperature? And second question is, if so, does variation in phenological advantage affect the competitive dynamics of species, right? And this is termed a seasonal priority effect. So there's the additional pieces of jargon you need to know. Now, just want to introduce you to the study system a little bit. We have a native species, honeywort or Cryptotaenia canadensis, that is depicted on the map in purple. It is a understory shrub that is kind of known for its competitive ability. And our second species is an introduced species from Eurasia, Dame's rocket or Hesperus nationalis. This is also a species that can, it's got a little bit of broader of a light requirement, so it can be in open meadows and forest edges, but also in the forest understory. You can see here on this map of the eastern United States, both species are pretty common throughout the northeast of the United States and southern Canada. And they are co-occurring at many species, so we think that they are probably competing quite often in the field, and we've seen that in our own personal observations as well. So now that we know them, talking a bit about the experiment. So the first experiment is we planted cohorts of these seeds in Petri dishes and exposed them to 10 different durations of cold stratification. So it's 0 to 13 weeks at 4 degrees. And then we moved them into 2 different incubation temperature chambers. So either 15 degrees or 20 degrees to really track to see this first question, how much does their phenology vary relative to each other? So here are the germination time courses. So what you have here on the x-axis is the day of the trial. And on the y-axis, we have the % germination of each species per day. So just to highlight a few of these panels. So under low stratification durations, so under no stratification, we see really different germination behavior. And that's not surprising for us because honeywort, our native species, we know has physiological dormancy and requires cold stratification, and Dame's rocket does not. So that's why we see, even under kind of without any cold stratification, it is germinating pretty well. What is perhaps more interesting about this study is that despite having these different germination syndromes, these different physiological requirements for germination, under cool incubation temperatures, so 15 degrees, and long stratification periods, you can see that their germination curves roughly converge. And that is interesting. So we took these raw data and we put them into a survival model, which is a time to event analysis that allows us to basically project the germination behavior beyond the 25 days of the trial. And so what you can see here, again, we have the weeks of cold stratification on the x-axis and the estimated days to 50% germination. If you kind of take a slice at 8 weeks of cold stratification, the invasive, depicted here in blue, is germinating somewhere on the order of 2 to 3 weeks before the native species. But when you get to higher levels of stratification, they're germinating at roughly the same time. So we suddenly have, based on climate alone, a version of this experiment or potential for the experiment where instead of planting the species at different time, we can expose them to different levels of stratification and generate this sort of phenological advantage we want to be able to assess their, to assess the priority effect, how much this matters. So this time, we did an experiment in a greenhouse where we planted these species in pots in competition. So there's both species are planted in each pot. And half the pots got 7 weeks of cold stratification and half the plots got 13 weeks. And then we tracked we let them grow for a season, so for about 3 months. And then we harvested the above ground biomass in each pot and used a metric called the relative growth rate differential to really understand competitive dynamics. So I'm going to talk a little bit about what we found. So what these models allow us to do is understand how much the density effect of the species, so adding another individual of each species, how that affects the overall compositional balance at the end of the season. So these density effects mean that positive values indicate that the final composition skews more towards the invasive Hesperus matrinalis, whereas negative values show that we're skewing the final biomass towards Cryptotania, the native species. So the first two effects are kind of unsurprising. If you add another seedling of Hesperus matrinalis, the pot starts to skew towards that species being dominant. If you add one individual of Cryptotania canadensis, the pot starts to skew towards Cryptotania dominance. And what is interesting is that sort of on a one-to-one basis, we're seeing more that Cryptotania is potentially a stronger competitor because one individual is skewing the pot more than, it's going more negative than the one individual of Hesperus that's positive. When we add the phenology effect, right, this priority effect, so this is telling us for every day that Hesperus matrinalis germinates before the native species honeywort, the pot starts to skew towards the Dame's rocket, Hesperus matrinalis dominance. And we can see that the effect of phenology here is stronger than the density effect. So when we project this forward into sort of the different parameter space, we see that in most combinations of density and time of phenological advantage, the Hesperus matrinalis or Dame's-Reich, the invasive, will end up dominating the pot. However, we can see that Hesperus matrinalis is able to compete when either it is at a higher relative density or that phenological advantage is low, so when they're germinating at the same time. So kind of some takeaways from this is we do see that phenological assembly can be highly variable for co-occurring species, and this is strongly dictated by climate. And then in turn, that these differences can really strongly affect competitive dynamics. And I think some of the next steps of this, while this is a sort of important case study by using climate to generate these effects, and that has some biological realism to it. Of course, this is a study that took place in a greenhouse with only 2 species. There are a lot of other factors that would really be needed to test it before we can kind of say this is happening in nature, but this is kind of an important proof of concept, laying the groundwork for moving forward. So I just want to, in the time I have left, just make some points about how this study potentially impacts our understanding of competition dynamics in the context of climate change and ecological restoration. So we found that cold stratification, these temperatures, are really the main driver of these dynamics. And while it's only 2 species, these are species that are sort of emblematic of natives, the comparison of native species with morphophysiological dormancy versus invasive species that don't have that dormancy requirement. So stratification temperatures are cool, not cold. So what that means is global warming could increase, decrease, or just shift these patterns of stratification. For example, if you are in a particularly cold region, warming might actually increase the amount of stratification that is happening in the environment as temperatures that were previously below freezing start to expand. And this is just a map from a study of changes in soil temperatures over the last century throughout Canada. And you can see these dynamics are variable in space, that in some places soil has gotten colder while other places warmer. So this suggests that kind of the way cold stratification dynamics are affecting species interactions is going to be spatially variable.
You're at 15 minutes.
Great, and I'll just end with putting this into the context of restoration. So we have this understanding of that potentially in thinking about phenological diversity in seed mixes could be a really important way of buffering against invasions. This is a study that found that from a pollinator context, restoration mixes that tend to lack early flowering species. And if that's true with flowering, that is probably also true with germinating. So really thinking about filling that early phenological niche is a good way to buffer against invasives. So that's some food for thought moving forward. And with that, I just want to thank institutions, Harvard and the Arnold Arboretum and the University of Massachusetts, where I've done this work, and I'm happy to hear some questions. Thank you.
Excellent. Thank you so much, Dan. There is one question waiting for you. John is actually asking for a copy of your slides. He sent his email. I will note though that this session is being recorded and will be available afterwards. If you feel that you missed any content, you can rewatch the presentation. But I'll leave it to you, Dan. If you wanted to connect with John, you can get his email address there.
Absolutely, thanks, John. I'm happy to share. And this is also I should have mentioned that this is currently in a manuscript that is in review and hopefully will be out for the public to read soon.
Excellent. No, that's great to know. If there are any other questions, please put them into the Q&A. I wanted to just mention that this was a very interesting study. And do you have any thoughts of doing kind of more multiple species rather than just 2, kind of making it more of a community?
Yeah, the original design had a lot more species in it, and thanks to our good old friend, the COVID-19 pandemic, the experiment got shut down for a bit, and it was harder to, get the seeds we needed after. So it ended up being kind of a 2-species case study. But I think, that would really seeing how other competitors modify these pairwise relationships would be a really important thing. Also thinking about competitions not just happening among seedlings in the nature, but thinking about, right, there are perennial plants that are also part of this dynamic, and how their phenology is also contributing to these dynamics would be kind of a clear next step for me, I think.
Absolutely. You answered my second question, was if you had planned on doing longer term studies as well.
Yeah, we are currently trying to use these studies to model longer term dynamics in a kind of process-based model, but that is, still the kinks are still being worked out of that. So I can't really report anything interesting or conclusive yet, other than it's harder than I thought it would be.
Absolutely. Lauren asks, in layman's terms, does variation in phenology advantage affect dynamics of species?
Yes. So, yes.
Competitive dynamics. Yes, sorry.
Yeah, we find that phenological advantage in the absence of risk, which is to say that there was no cost for our invasive. There was no potential to be frozen by a late season frost or anything. So, but in terms of these kind of within season dynamics, yes, we find that phenological advantage, the more that species germinated before its competitor, the stronger its competitive outcome was.
Excellent. Okay, thank you so much, Dan. We're going to move on to the next presentation. If there's any other questions, feel free to still pop them into the Q&A, and Dan, you can feel free to just type out those answers. Thank you so much. Okay, to close off this session, I'd like to introduce Pedro Antunes from Algoma University to talk about the vulnerability of non-native plants to novel pathogen attack. Do traits matter? And Pedro, feel free to join us. I see you already have your presentation shared, so that's excellent. Take it away.
Hi, Larissa. Thank you. Can you hear me okay?
Yes, you sound great.
Perfect. Thank you so much. So yes, I'm going to talk about the vulnerability of non-native invasive plants to novel pathogen attack and explore a little more whether or not plant traits matter. So, we've all seen this curve of the pool of introduced non-native species. A large majority, they do not establish. Of those that establish, a small portion become invasive and then spread in the introduced range. Over time, the interactions with novel pathogens start to occur in the introduced range, and they may cause the decline of some invaders eventually. So understanding the traits that may contribute to pathogen vulnerability is then important, or it's an important goal to estimate the long-term consequences of plant invasions. So here you see, for example, if you have a leaf that has low specific leaf area, so it's very thin but very, very wide. You may have a very large surface area for pathogens to come in, for potential enemies to interact with this plant. But if you have like a needle, that specific leaf area is different, right? It's a very much lower surface area, and maybe a plant that has traits more akin to this, just of course, thinking about leaves, which is very narrow, this is just an example, may be more persistent to invasion. So over the last decade, there's increasing evidence suggesting that pathogens have the potential to regulate the long-term dynamics of invasive plants through the emergence of novel interactions with pathogens that are capable of reducing the invader's fitness. So pathogen accumulation. So here you can see the results from a study by Bernd Pelossi's group showing population growth rate of garlic mustard over time. So the points represent the mean growth rate across sites. The lines and shaded areas represent marginal means, and with 95% confidence intervals based on, it's a linear mixed model. And but what's clear here is that the longer population is present at a site, the smaller the growth rate. So then the question becomes, could this be mediated by plant pathogen interactions? So we have known for over 10 years that negative feedback actually correlates with the years since invasive plant establishment. And that was clear in this study by Diaz, looking at where you see years since establishment for different invasive plants and in New Zealand, and how the feedback has become more and more negative for each one of these plants for the longer they've been established. But how common then? So the question was, how common is pathogen-mediated population declines among invasive plants, and how do these interactions actually change over time? So that's one of the questions that we've had in our lab for quite some time. So we published a paper in 2019, and where we used next generation sequencing, and we looked at the roots of all the plants that are present in were present in an abandoned agricultural field site in Ontario, actually close to Guelph. And so we used next generation sequencing on these roots, and we identified fungi present inside the roots of all the plants that were present in this place and at this field. And then we used the database, Fungild, to assign how functional, actually the known functional roles of fungi, including obviously pathogens. And so you see a phylogenetic tree here and the pictures of all the plants that are present in an old field, in this particular old field. And the orange ones are exotic species and the green ones are native species, mostly, unfortunately, are most species are exotic. They are not invasive. They are exotic, and I think this is an important thing to think about at this stage here in the talk. So, what did we find? So, actually, contrary to what we expected, there weren't distinct differences in fungal community composition between exotics and native plants, including pathogens. So basically what this suggests long residence times and obviously meaning potentially that the consequent naturalization of exotic species in the region. So pathogens are present in exotic species, and this may help stabilize their populations in this old field community. So then the key question becomes, can we make predictions about which invasive plants are most vulnerable to pathogen-related declines? So as these novel interactions occur, we have to think about this. But it's also important to note that while pathogen accumulation may not undo the socioeconomic and environmental damage caused by plant invaders, right? So we consider that better knowledge of such processes could potentially lead to novel ecological applications tied to prevention, management of plant invasions. So I wanted to make that note there. So basically, we use the TRI database, the trait database that's available, and we looked at plant functional traits, which are defined as morphophysiological traits that impact the fitness indirectly through effects on growth, reproduction, survival. So we first created these trait categories you see here that we considered are likely to determine the resistance and tolerance of plants to pathogen attack. And so here in this illustration, you see different traits that could be linked to pathogen attacks. So architecture and growth rate, secondary metabolites, the seeds, traits tied to flowers, fruits, all of that. So then for each trait, we looked for evidence that it can be involved. So actual evidence that it can be involved in pathogen resistance and or tolerance. For example, for stomata, it has been shown that lower stomatal density in leaves can prevent pathogen infection, right? So also stomatal closure in response to pathogen presence is going to reduce infection. So if the stomata, the smaller, there's less points of entry specifically. And then, of course, for all of these traits, we looked for evidence and we have the references listed here. So in this particular case of stomata, resistance more than tolerance was important. So next, we looked at for evidence of novel pathogen-invader interactions, knowing that there was no synthesis of documented examples of novel pathogen attack of invasive plants. So to determine the prevalence of novel pathogen interactions, we compiled data from on plant invader diseases using these search terms you see here. And actually, this search then produced 661 results. And but there were a few conditions we had, and so we looked at all these articles one by one, and our conditions was that the plant species must be considered non-native invasive. Putative pathogens must have been found in the non-native range of the invader. The effect of the putative pathogens must have been tested by inoculating the invader with the pathogen, of course, like Koch postulates in microbiology. The pathogen must be native to the non-native range, so basically a new encounter. So we identified 31 invasive plant pathogen combinations with 31 invasive plant species experiencing novel pathogen effects from 21 unique pathogens. And this was reported in this vast, this large table. So the invasive plants came from a variety of functional groups, including forbs, there were 14, shrubs, 6, grasses, 4, legumes, 4, aquatic plants, 2, there was 1 tree only. So 20 were from North America, 4 from Australia, 3 from Europe, 3 from Africa, and 2 from Asia. So we recorded the approximate year of introduction as well. And then we looked at whether or not the pathogens were observed on the invader from the CABI database or references. So actually the amount of time for disease to develop on the invaders range was around between anywhere from 15 years to 250 years. The average was 120 years. But I mean, the main point in this whole talk is that these data are very limited. Here you see what we found, for example, for garlic mustard that I'm highlighting there because it's kind of difficult to see. But there was one pathogen that, under greenhouse conditions, under laboratory conditions, could actually have up to 100% mortality. So we then used the data collected on pathogen impacts that were shown in the previous table, and we applied a multivariate model to associate plant traits with pathogen effects. So for this, we actually compiled data from the TRI database for each invader, and then we selected all the traits. So these here were available for 75% of the species. And you can see at the end of the arrows, they include plant height, nitrogen, leaf nitrogen content, seed mass, specific leaf area, and leaf dry matter contents. So we calculated the pathogen effect as the relative difference in fitness performance. So for example, survival, reproductive output between the control and pathogen infected plants averaged across all available studies of each plant pathogen combination. So from the very limited data, we were actually to pick up a signal and were able to collect these traits and indicating that they can be useful in determining pathogen effects when included, obviously, as part of a broader framework. Many of the traits identified as important, they were not available for the invaders we identified, which makes it clear that more data is actually needed here to improve the generality of the model. So as we found evidence for 31 cases of invasive plant associations, like I mentioned already, with novel pathogens, but we expect that there are additional cases that remain identified due to the limited research that exists. So in conclusion, from this framework, we can begin to determine the trait axes that are most associated with pathogen vulnerability. So we found that together, plant height, nitrogen leaf content, seed mass, specific leaf area, and leaf dry matter content can be useful in determining pathogen effects when included as part of a wider or a broader framework. more data for invasive plants are needed to keep refining the predictive capacity of this framework. Understanding of invasive plant pathogen interactions can lead to breakthroughs at both fundamental and management decision levels. If we know what plants may be more prone to pathogen attack, maybe they may not be prioritized as quickly if there's a lot, and we know we have a lot to deal with. But this, I mean, obviously this is requires a lot more thinking and consideration. But for sure we can conclude is that we need much better monitoring. And when we are reporting locations of invasive plants, for example, through EdMaps or other resources, the reporting of disease in invasion plants, either above ground, below ground, is I think can be very, very useful. So I'd like to acknowledge this work is published and it's available in Biological Invasions. I'd like to acknowledge my two collaborators in the lab that worked with me, Catherine Fahey, who did amazing work with this, and Dr. Akihiro Koyama, who also worked in the Journal of Ecology paper I showed. And of course, my funders, the Canada Research Chairs Program and NSERC. And with that, I'll take questions. Thank you.
Thanks, Pedro. That was very interesting. It's actually really nice to also hear about your research now after 7 years of being your thesis undergraduate. No, I'm wondering if you have, is there somewhere where people can send samples for someone to look at what pathogens might exist on those populations of invasive plants that they're finding on the landscape?
That's a really good question. So we actually started at some point a project. We require a lot more funding for it, but where we started to have the opportunity for people anywhere and colleagues to collect plants, specific plants we were interested in, specific invasive plants, and they could collect roots and actually send us the roots. We would look at roots, looking for lesions, for evidence of fungal pathogens in these plants, and then we would know the location where the samples were collected, the age of the invasion or the populations, and for how long they have been there, so that we could continue, and of course we would know all the traits. So we had that project going. I think it's very needed. So maybe it's something that we could explore with collaborators or as part of a much bigger consortium. But yeah, that would be great.
That's awesome. I'm glad that it at least was started kind of as a pilot. So hoping to see if that can expand.
Yes, the pilot made me realize how big that can get. Yes.
Absolutely. I think that's the case every single time for any new project. We do have a question from Leonardo. Have you thought about the opposite scenario? So invasives being a reservoir of pathogens that can cause disease on natives.
Yes, we have. We actually have a few studies published with, for example, with garlic mustard. Yes, for example, and also one with dog strangling vine, where we actually, it's available. I think it's Biological Invasions. I can share the paper later, but yes, the plant actually with dog strangling vine, we were able to isolate fungal pathogens or putative pathogens from this plant, and actually found these putative pathogens were actually either neutral or beneficial on the host, and potentially they could be pathogens on native plants. So yes, that's the spillover hypothesis is definitely something that we are looking into as well, and something that deserves more attention.
Awesome. And Pedro's email is in the chat if you are looking to get a hold of him and looking for any of those papers as well. Nadia asks, is anyone analyzing herbarium specimen images for visual cues of disease in invasive plants to further this research?
Not that I know of. We also have a paper on how we can use plants from herbarium data to better assess lag times, predict lag times on invasive species curves, and so there's a conceptual framework there. I'm also happy to share that. You can find those papers on our lab website. Adding pathogens and pathogen or enemy attack, it could be insects, could be anything, to look exploring herbarium records. That's a great idea, and it's something that yes, would be important work to be done, and could contribute to build a wider framework that we proposed. Yeah.
I love identifying gaps. Anyone interested in listening? You're up.
Somebody
Deb,
can come and do some grad work with us, or yes, we could look at that.
Exactly. Deb wants to know, does EdMaps currently house a data set where this can be documented and it just needs to be used more? Or is it a layer that could be added?
You know, that's a good question. I don't want to comment because I use EdMaps and for different purposes, but I haven't really explored lately. And I know they're always adding new functions, and I think it's a wonderful tool. So I at this point, I don't know.
I think that it definitely could be something that would be added, or a layer added, but would need to look into it a bit further. Yeah. Okay, excellent. Thank you so much, Pedro. That's all the time we have for today. If anyone, again, has any more questions, please, his email is in the chat. Feel free to reach out to him. I'm just going to share my screen. Okay, so one last thing before we close out this session, I'd like to invite you all to participate in the Invasive Species Awareness Week, or ISAW, taking place from February 20th to 26th. You can get involved by liking and sharing or retweeting posts created by participating organizations, or also creating your own posts and using the hashtag Invasive Species Week during ISAW. Visit our website to see the online toolkit, ready to use materials and resources there for your organization. And with that, I will pass it back to Mackenzie.
Thank you so much, Jersa, for taking us through a great first half of the morning. And thanks to all of our speakers that joined us this morning. Special shout out to Pedro, who is one of our Invasive Species Center Leadership Award winners last night as well. So thanks for joining us today again, Pedro. We're going to take a quick 10-minute break before we get started with the rest of the morning and our second session. Jersa will be back with us for the rest of the morning, so we will see you back in the next session at 10:30. For anybody who just joined us for this session and missed this morning, we're also having our invasive species trivia at lunchtime today. All you have to do is join the trivia session and have your mobile device handy, and you can join us to showcase your invasive species knowledge. So thanks everyone for joining, and